Thank you all for coming and making it out. So I think the two talks before really set my talk up really well, because we have these universal principles of life, and now we're kind of constraining the metabolism of single cell, and now I'm going to take you into the environment where it gets really messy really fast. So I work on understanding how microbes are interacting and interfacing with themselves and their environment. And the model system that I use are stromatolites here. And stromatolites have popped up quite a few times over the course of this week. And so, but I thought I would define the term because I think of them as ecosystems, but really stromatolites are structures. They're sedimentary structures that are made by the activities of microbes. And through their actions of trapping and binding sediments from their environment, they are, and also through precipitation, they have this very iterative growth process that creates these laminations or layers of carbonate structures that we think of as the stromatolite structure. And we've heard many reasons why stromatolites are really interesting studies to look at. They can be important reservoirs for biosignatures, analogs for maybe the carbonates on Mars. So these are just some of the topics, but they're really productive ecosystems. John Spear the other day called them microbomats little rainforests, and you can really apply some of these ideas about microbes interfacing with the environment on these ecosystems. And so I work on a community there's several different stromatolites across the globe, but I work on the ones in Hamlin Pool in Shark Bay, Western Australia. Shark Bay is a UNESCO World Heritage Site, and Hamlin Pool is kind of cut off from the rest of Shark Bay by this location. It's called a 4A sill that kind of restricts water flow and creates a hypersaline environment. And these stromatolites were discovered in the 1950s, and there have been these kind of canonical mat types that have been described over the last few decades, and some of them form stromatolites, and some do not. And these names have been around for decades, but the location of where they are is a major driver of what type of microbes and how they're making stromatolites. And so I'm going to talk about these four mat types in my analysis, and the pustular forming stromatolites, I did not name that, but the pustular ones are in the intertidal zone where the smooth and coliform seem to be always found in the subtidal. And then you have the non-lithifying mats, which are definitely in the upper intertidal regions. And so the questions that I'm looking at is are there really footprints or metabolic footprints for each of these mat types? And I'm pretty much comparing the non-lithifying to the lithifying systems. And are they dependent on their geographical location or other aspects of their environment? And so again, I'm looking at collecting samples from these different stromatolites and non-lithifying microbial mats from two different locations within the pool, a more northern site that is more less saline, and a more hypersaline place called Go Point in the southern part. And we've looked at the microbial diversity, we've looked at the metagenomics, and now I'm going to talk a little bit about applying a metabolomic approach to these ecosystems. And just all the data I'm talking about was collected at noon, the peak of photosynthesis. And so unfortunately, most of the data that we get back are unknowns. And we're always kind of working on trying to improve that. But the known communities, and I've made two plots here, one with just the knowns and with the unknowns. So even though we don't know what most of these metabolites are, they seem to be, we still see very discrete populations of communities, suggesting that each of these mat types have a really distinctive fingerprint of metabolic, or a pattern of metabolic molecules in their profile. And they follow a very discrete path with the seaward, the most deep water microbialites, and then there's also the non-lithifying. So let me just add this on there. So whether or not you have unknown or known, you're getting very discrete populations of the metabolic profiles that are really clearly separated by whether the mat type, but we are also seeing a geographic component. So there's two things driving the differences in these communities. And so we can see really distinct patterns. So these are just the top, we're just dealing with the known metabolites at this point, and we're seeing really distinctive patterns within these mat types. And these are the top 50 metabolites that we're recovering from these mat types, and they're divided up based on mat type and location. But we're seeing two major trends here. Some patterns of molecules really are affected by water depth, and we can really distinguish and tell you whether or not that mat type is intertidal or subtidal. And then we're also seeing pronounced differences of whether or not they're lithifying or not lithifying. So it really depends on what your question is, but we can also see this group of clusters only seems to be appearing in lithifying stromatolite-forming mats as opposed to the non-lithifying. So I'm going to give you one specific example of a comparison, a pairwise comparison between a stromatolite-forming mat and a non-lithifying mat to see what we see. And so this is a volcano plot, plotting all of the metabolites that we're comparing in these two mat types. And on how you read this is everything on this side is enriched or up in the stromatolite-forming mat, and everything on this side here is up in the non-lithifying mat and down in the stromatolite mat. And everything in blue and red here are significant, and everything in red is a twofold change or higher. So there are lots of different metabolites I could talk about, but one of the examples is they seem to have, the stromatolite-forming mats and the non-lithifying mats seem to have very different strategies of how they might deal with osmoprotectants. For example, one of the most dominant molecules in the stromatolite-forming mats is a molecule called trigonelline, and that is an osmoprotectant, but it's also an important bacterial regulator. The non-lithifying mats do not seem to have this molecule. They seem to be more enriched in other types of osmoprotectants, in this case, betaine. And the carboxylic acid profiles seem really different between mats that make stromatolites and mats that do not. A good example here, carboxylic acids are important in carbonate morphology. They're not necessarily serving as nucleation points, but they can actually control the type of morphology that you see. In the case of malic acid, which is strongly enriched in the stromatolite-forming mats, they can actually absorb onto the calcium carbonate structure, and actually malic acid specifically makes dumbbell-shaped carbonate structures. So you can see that was enriched there, but the non-lithifying mats do have carboxylic acids, they need them, but they have very different profiles, and in case gluconic acid is associated often with carbonate dissolution, actually. So by teasing apart, we can actually really see distinct profiles between what makes a stromatolite different from a non-stromatolite-forming mats. And now what we're doing is now we're taking that metabolomic data and overlaying it with some of the metagenomic data that we've accumulated. And we have about 35 different metagenomes for these communities. And we're trying to now, there's going to be a lot of organisms that make malic acid, and we're trying to associate with, this is a Pearson correlation of a smooth stromatolite-forming mat with a pustular, and we can now see taxonomic differences of who might be making those different metabolites. And we're taking it a step further and also looking at what pathways are enriched and not enriched, depending on what you're looking at. And in the case of the intertidal, here's a case where we're comparing an intertidal stromatolite forming mat with a smooth forming mat that's in the subtidal, and we're seeing really differences in what type of metabolisms are used, are enriched in those communities. So in the case here of the pustular forming mat, we're actually seeing strong enrichments. Everything in green here is a photosynthesis associated mat. So when you're in the intertidal zone, those stromatolite forming mats tend to be more photosynthesis driven. Whereas in the subtidal, we're seeing many more like sulfate reduction, more heterotrophic metabolisms driving that. So we're really seeing, we're getting profiles not only of basically what metabolisms are there, but maybe some of the major processes that are forming these ecosystems. And now we're layering in the environment. into this. Over the last 6-7 years, we've had data loggers all throughout the different parts of the pool here, capturing temperature, salinity, and tidal heights for all these environments. And my colleagues at University of Miami have created a really detailed bathymetry map and captured some of the more of the macro features of these stromatolites. And we're starting to layer in the environmental data associated with these communities. And one story I'll tell you is about salinity. Salinity in a lot of environments drives certain microbial populations. I've seen it in the mats in the Bahamas, but in this particular environment, salinity accounts for very little of the diversity that we're seeing in the communities. Those really tight, discrete patterns that we saw in looking at the metabolome kind of gets lost when we start just focusing on salinity, and only about less than 10% of the variation could be explained by salinity. So some environmental factors like water depth seem to be more important than other factors like salinity, and we're slowly teasing those interactions apart. And just to kind of recap some of these big ideas, are there different distinct metabolic signatures with each of these mat types? Yes. There does seem to be stromatolite-specific metabolites that are found, and yet you can also then use other patterns within the metabolome profile to understand where they are or what signature they have that's dependent on the environment. And one thing I didn't really talk about are the discrete, really different surface features of these morphological features of the stromatolites, and what the big factor seems to be is where they are in the water column, and that really impacts the genetic content of these systems. And now what we're trying to do is correlate those surface mat features with the subsurface structures to say what might be the underlying carbonate structures in those communities. And I just want to say thank you to Giovanni Babaloni. She was the graduate student who did a lot of this work, and she's now a postdoc at the Los Alamos National Lab. And Pam Reed, who has been a big help with the geological aspects of this, Tim Garrett, who helps with the Metabolomics, and Bush Heritage and Western Australia government for allowing me access to this UNESCO, really important UNESCO World Heritage Site. And Phil Playford, who discovered these in 1952, he passed away about a little bit more than a year ago. So I really want to say thank you, and also thank you to my funding sources. So that was just a big picture to kind of give you an idea of how we're applying these systems biology approaches to modern living stromatolites. Thank you. Not bad, only 30 seconds over. Not bad.
All right, thank you, Jamie. One quick question.
Yes. Oh. Hi.
Do you have any chance to do any sort of mesocosm experiments to start varying those values?
Yeah, actually, I've taken some of these mats into the lab and built little environmental chambers where we can manipulate. And interestingly, I've manipulated CO2, salinity, and temperature on these. And it's really hard to break a stromatolite, let's put it that way. These systems, you can manipulate, and obviously they've been around for a long, with very dynamic environmental conditions. And we have been able to track and follow how the community is changing under these manipulations. And we can get these stromatolites to form and grow under lab conditions. So we're doing that. Thank you. All right.
One more maybe?
Nope.
That's all right.